And we have two ends, anterior end head present at the anterior end, and in case of animals, anterior end transformed into head. The head formation process is known as cephalization. We know cephalization means head formation. All sensory organs in most of the living animals are concentrated at the anterior end, and in case of vertebrate, there is cephalization, higher level of cephalization, which indicate that these are highly evolved animal, and high degree of cephalization and brain formation is present in case of primates. We can equally divide the animals into two equal halves from sagittal plane, whereas coronal plane, this plane cannot divide the animal into equal halves. Then there is transverse plane, and only sagittal plane can divide the animal into two equal halves. Body tissues, on the basis of body tissue formation, we have two types of animals, diploblastic and triploblastic animal. We were talking about body tissues and only one group of phylum Porifera which have no true tissue formations, while rest of the eumetazoans have body tissues, and all these body tissues made up of different germ layers. Among these germs, there's endoderm, ectoderm, mesoderms. These are the three different layers. In case of ectoderm, this is outermost covering in case of animals, and it gives rise skin and integuments, whereas second layer, which is known as endoderm, this is innermost layer, and this layer give rise digestive system, liver, and lungs, other associated organs. We have two types of animals on the basis of germ layer, diploblast and triploblast. In case of triploblast, we have three layers: endoderm, ectoderm, and mesoderm. You can observe in these two diagrams. You can observe in this diagram, this is outermost layer, which is ectoderm. Body tissues in animals, true tissues, are a collection of specialized and cells isolated from the other tissues by different membranous layers which were developed during the course of development. Only if we have sponges which are lacking true tissues, while rest of the animals are all having true tissues. And on the basis of these germinal layers, we can divide the animals into different groups like bilateria and radiata. Bilateria are those organisms which can divided into two equal halves from any plane. And second is radiata, which are mainly triploblastic animal, and all these animals are made up of three layers. Two types of animal, diploblastic and triploblastic animals. Diploblastic animals are generally have radial body symmetry, whereas bilateria, which include the animals which have bilateral symmetry and made up of three germinal layers. In this slide, you can study most of the triploblastic animal have body cavity, are known as coelomate, and some of them which have lacking body cavity, and such animals are known as acoelomate. In case of true coelomates, they have well-developed body cavity. This is the region uh, which is known as coelom, and this coelom is present in between the two mesodermal layers. Red color is reflecting mesoderm, whereas inner layer, which is known as a gastroderm, representing by yellow color, whereas blue color is an ectoderm. In first case, acoelomate, there is no body cavity, and that's why these animals are known as acoelomate. These animals are represented by platyhelminthes, coelomates from mollusca to arthropoda, and all deuteriostomes, including chordates, echinoderms, are coelomate organism. In these organisms, true body cavity is present, which is known as coelom, internal digestive tract, endoderm, and this is the external part of mesoderm, and you can say, and this is internal part of mesoderm, which is associated with the digestive system. So in case of pseudocoelomates, they have coelom, but this pseudocoelom is present between digestive tract and mesoderm. If you look inside the mesoderm, this cavity is known as pseudocoelom. Pseudocoelom is fully functional body cavity, and in earlier literature it was named as false coelom. But later on, this name was retained as such, but this is not false. Proteostomes and deuteriostomes, these groups are divided on the basis of animals' development. Proteostomes and deuteriostomes. Triploblast mostly have two different patterns of their development. Proteostomes and deuteriostomes, basically, we will study the various, you can observe the cleavage. In case of cleavage, you can study, this is 8-cell stage during the development. In case of proteostomes, you can observe the pattern of cell division is spiral. In case of deuteriostomes, there is a radial division. Radial division, you can observe these cells are placed at the same spiral division is determinate. It means these cells are already, and indeterminate cells, these indeterminate cells, you can say. In case of cleavage, we have two different types of cleavage, spiral cleavage and radial cleavage. Spiral and determinate cleavage, these cells are spirally arranged. And if we separate all these cells from each other, no one can successfully transform into an independent individual. Whereas this 8-cell structure, if we separate all these 8 cells, each cell have the potential to convert into the young one. These cells are indeterminate, whereas these cells are determinate. If we separate all these cells from each other, then what will happen? There will be no mature or adult individual formation. Then second point is the coelom formation. Coelom formation, a process in which coelom is developed. This coelom developmental process starts after reorganization of cells during gastrulation. In case of proteostomes, mesoderm develop from the region near the blastopore, whereas in case of deuteriostomes, development of mesoderm starts from the roof of orchantodon. State of blastopore, in case of proteostomes, mouth is developed from blastopore. Whereas in case of deuteriostomes, blastopore develop into an anus. Now we are moving towards the next experiment, how we can determine whether these cells are having potential to convert into a mature individual. How these individuals can convert into the mature individual. For this purpose, indeterminate cleavage in which division occur, and if we separate these two cells from each other, cell has potential to convert into a separate individual. Whereas determinate cleavage after first cell division, if we separate all these cells from each other, and no one cell will convert into an adult individual. Proteostomes consist of spiralians and ecdysozoans. Spiralian animals grow by gradual addition of mass to the body and undergo spiral cleavage. And there are two main groups of spiralians, Lophotrochozoa and Platyzoa. Proteostomes, trochophore larvae present in mollusca and annelida, and Lophophore larvae present in Bryozoa, Brachypoda, and Pterobrachia. These are the smallest phyla which are present in invertebrate. These animals are collectively known as spiralians. Whereas the second group is Ecdysozoa. Ecdysozoa includes those animals which molt during their development, shed off their skin during their development. Platyzoa are acoelomate, and their movement occur through ciliary action. Ecdysozoans are the animals which perform the function of molting for their development during their life history. And among this group, arthropoda and nematode are present. And arthropod is one of the biggest group in animal kingdom. Moving towards the next slide. Second group is deuteriostomes. Deuteriostomes include chordates and echinoderms. These animals have radial cleavage and indeterminate cleavage. I'm repeating once again, indeterminate cleavage is that cleavage if we divide the zygote into 8 cells and separate all these cells, and every cell will convert into an independent individual. It means their fate or their future is not determined yet. So that's why we call these animals indeterminate. Deuteriostome consists of few phyla. Among these phyla, we have echinodermate, hemichordate, phallochordates, tunicate, and vertebrata. All these phyla have a skeleton which is developed from the mesoderm. Echinoderms, these animals are sea stars and exclusively present in marine environment, whereas we have chordate. Chordata is that phylum which is highly developed among the all organisms. There is well-defined and regular pattern of, you can say, evolution among the chordates. The process of evolution you can observe during the course of evolution among chordates. We will start from invertebrate. Invertebrates accounted for 95% of known animal kingdom, and among these invertebrate, more than 70% individuals are belonging to phylum Arthropoda. They occupy almost every habitat on this planet, from boiling water to the deep sea hydrothermal vents in Antarctica and other regions. These invertebrate has adapted a variety of habitat. Some of the insects are flying, some animals are on the earth's surface, some animals are burrowing and are living in fresh water, and most of them marine mollusks, echinoderms, and some of the other phyla are living in marine environment. First phylum, that is the that belong to simplest organism, that is phylum Porifera. We include phylum Porifera in Parazoa. Animals belonging to Parazoa, they have simplest body, and there is no tissue formation. Further, in case of phylum Porifera, why we name it Porifera? Porifera mean pores. Because of that, sponges have pore on their body. These sponges these sponges have pore on their body, and water enter into such sponges have pores on their body, and water enter into their body through the pores, and these pores are known as ostia. Water enter into the body and then move out through the main opening, which is known as osculum. You can observe osculums in these sponge structures. And there are two types of cells which are cells, two types, flattened cells. These cells are known as choanocytes, and other cells which are collared cells and present inside the body. These are known as collared cells. And there are several types of cells which are present in between these two layers, amoebocyte cells, and some of cells are present inside the sponges, which are meant for the formation of calcium. Calcium carbonate skeleton and silica spicules. Spicules are the spine-like structures which are present in sponges. Sponge's skeleton is made up of spicule and fibers, and all these fibers are known as spongin fibers, and all these are combinedly form skeleton in case of Porifera. Porifera is one of the simplest group, and it can display maximum regeneration. Maximum regeneration इनके केस में होगी अगर आप स्पॉन्जेस का कीमा करके उसको बारीक कपड़े से छान लें और उसमें से सेल को एक एक सेल को अलग कर दें और फिर उसको पानी में रख दें, तो वो सेल्स एग्रीगेट करके दोबारा आप क्या बना लेंगे, एक स्पॉन्ज बना लेंगे. That is the great ability of sponges to reproduce. Sponges reproduce by means of you can say fragmentation, budding, and these are the type of asexual reproduction. And some of sponges reproduce sexually. By means of sexual reproduction, some sperms from other sponge enter through the ostia and come in the body and retain within the body and fertilize the egg. And after decay and decomposition of sponge, that zygote release and convert into a free swimming larva that is known as amphiblastula. उसको हम क्या कहते हैं? Amphiblastula. So on the basis of spicules and exoskeleton of Porifera, we can divide this phylum into different classes: Calcarea and Silicia. These are the different classes of sponges. In case of Eumetazoa, these are the animals which have true tissues in their body. Phylum Cnidaria or Coelenterata, there are more than 10,000 species which are living in marine ecosystem, and some of them freshwater. These animals which belong to Eumetazoa phylum Cnidaria are diploblastic, and there is radial symmetry. Radial symmetry indicate that these animals are primitive, and there is no reproductive system, no circulatory system, no digestive system, no excretory system, and no respiratory system. And there is a digestive system that is of primitive type. Their digestive system is just like sac-like digestive system, which have one opening which act as mouth as well as anus. Such digestive systems are known as sac-like digestive system. So in case of cnidarians, there is no central nervous system. However, there is a network of neurons present in the whole body of animals, which help the animal in coordination. Such type of nervous system which have no central brain or central aggregation of neuron, those type of nervous system are known as diffused nervous system. Cnidarians are carnivore in nature, and these animals feed on fishes, crustaceans, and other kind of animals. Tentacles of these cnidarians are loaded with nematocytes. These nematocytes are the cells whenever any animal touch with the trigger of this animal, it will puncture the skin of animal and inject the poison within the body. And this is the so small amount of poison which is injected by the hydra and other animals belonging to the phylum Coelenterata. It is highly poisonous, and it immediately paralyzes the prey, and these tentacles move the animal into the gastrovascular cavity, where digestion of animal will start. And in the case of cnidarians, there are two types of body. One of them, polyp, which is attached to the substratum. And polyps are small buds, and these buds convert into jellyfishes, which are known as medusa. These jellyfishes are basically a free-swimming individual, and after gaining maturity, these medusae will produce eggs and sperms and both eggs and sperms fertilize each other, and after their fertilization, formation of zygote and their larva, which will produce as a result of division within the zygote, is known as planula. So in case of sponges, larva is amphiblastula. In case of cnidarian, this is planula, and we have coelentrates के केस में हाइड्रा में हाइड्रोस्टैटिक skeleton होता है. However, in case of anthozoa, sea anemone, and corals, sea fans, we have calcareous exoskeleton, which help the organism to form coral reefs. You can observe coral reef in tropical oceans, and several islands are made up of coral reef. Hydrozoa, this class consists of Hydra, Portuguese, man of war, three classes of coelentrates, Anthozoa, Hydrozoa, and Scyphozoa. Next category is Bilateria. Bilateria is characterized by the bilateral symmetry. In this diagram, you can observe bilateria. We can divide the bilateria into proteostomes and deuteriostomes. Proteostomes further divided into Lophotrochozoa and Ecdysozoa, deuteriostomes into echinoderms and cardates.
सर सूडोसिलोम वाला क्वेश्चन के अब जब रिसर्चेस हो चुकी हैं कि उनके अंदर एक ऑर्गेनाइज्ड सिलोम है, तो फिर उनको सूडोसिलोम क्यों कहते हैं?
बेटा वो उसका नाम उसको रिटेन रखा गया है क्योंकि वो ज्यादातर लिटरेचर में अब वो नाम आ चुका है. तो आप समझते हैं ना, कुछ चीजें ऐसी होती हैं जो गलतुल आम होती हैं और उनको उसी तरह एक्सेप्ट कर लिया जाता है.
सर जो ह्यूमन की बॉडी होती है वो बायलेटरल होती है या रेडियल होती है? ये
बायलेटरल सिमेट्री होती है ह्यूमन की बॉडी में.
और सर जो ट्रिप्लोब्लास्टिक होते हैं वो सारे रेडियल होते हैं?
ट्रिप्लोब्लास्टिक इनमें बायलेटरल सिमेट्री होती है. तमाम ट्रिप्लोब्लास्टिक में एक फाइलम ऐसा है अकाइनोडर्मेटा, उसमें रेडियल सिमेट्री है, लेकिन उसकी डेवलपमेंटल स्टेजेस देखें तो उसके लार्वी में क्या है, उसमें भी bilateral symmetry होती है. Or those animals which are included in invertebrate, while rest of these four are included in chordata. Hemichordata, Sphelochordata, Urochordata. Urochordates are known as tunicates, and vertebrata. These are the subphylum of phylum Chordata, which we will study later on in detail. In all these deuteriostomes have radial symmetry and indeterminate cleavage, and their blastopore develop into anus at the early developmental stages.